Hi friends, welcome back to my channel. In previous class we discussed about the what is meant by dipeptide, tripeptide, tetrapeptide and polypeptides. Also we discussed about the types of the proteins, fibrous proteins, also globular proteins. Today in this session we will discuss about the what is the structure and shape of the proteins. Okay, what is the structure and shape of the proteins? But do you know what is the structure of the proteins? It is a very complex structure. That means we cannot easily study the structure of the proteins. Everyone should remember the structure of the proteins is complex. It is very difficult to understand the structure. So that's why the structure of the proteins we can study at 4 different levels. What are the 4 different levels? One is the primary level, that is called as a primary structure. Second one, secondary structure. Third one, tertiary structure. Fourth one, quaternary structure. See here 4 different levels are there. So when you are going from one level to another level, you will get a difficulty. Suppose we are playing one game. In this game different levels are there. First level, second level, third level, like this levels are there. We are playing the game. Do you know that levels? In first level it is always easiest level. If you are going second level means it is very difficult to compare to first level. If you are going third level, it is very difficult to compare to first and second levels. Like that also in proteins there are different types of structures are there. Primary structure, secondary structure, tertiary structure and quaternary structure. If you are going from primary to secondary, it is very difficult. Okay, we are going secondary to tertiary means it is very difficult structure compared to secondary. If you are going quaternary structure means what happens, it is very difficult to understand compared to remaining structures. That's why it is easiest way. If you remember all this primary, secondary, tertiary means you can easily understand what is the structure of that proteins. Okay, if you are seeing the protein structure, there is a complex. That's why the structure of the proteins we are studying at 4 different levels. First one we are starting from easiest level and we are going to the difficult level. Now I will explain what is the structure of the proteins. We are starting from the primary level. In this one, the first one is primary structure. Actually this primary structure explains about the sequence of amino acids. The sequence of the amino acids in a polypeptide chain. Polypeptide chain. So everyone should remember. It explains about mainly what is the sequence of the amino acids. Suppose I will take one of the polypeptide. I will take one polypeptide, one example. I will write one general formula A1. It is bonded with A2. It is bonded with A3 and so on AN is there. Here A1, A2 are amino acids. That are the monomers. Monomers are combined, they form a polymer. This is called as a polypeptide chain. In this polypeptide chain, there are many monomers are there. Many monomers, that monomers are amino acids. Okay, actually primary structure explain about the sequence of the amino acids. It is one of the sequence. Okay, suppose I will change the sequence. Here A1 and A2 like this one sequence is. Suppose I will change that position of the A3 to A2 place. A2 place to A3. Suppose I will exchange that the position of that amino acids. Now you will get a one more sequence A1, A3, A2 and so on AAN. Do you know this is another sequence? So because of the another sequence, what we will get? Another protein you will get. This is the one of the polypeptide chain. It is also one of the polypeptide chain. Okay. Whenever change the sequence, you will get another polypeptide chain. It is also polypeptide. It is also polypeptide chain, but it is different from this first one. Why it is different? Because the position of the amino acids changes. So because of that, both are different proteins. Okay, the polypeptide chains are called as proteins. In this polypeptide chain, it is one of the sequence. In this polypeptide chain, another sequence is there. So different sequence means that are compulsory different proteins. Okay, this is about the primary structure. It explains mainly about the sequence. If you are changing the sequence of the amino acids, it creates the new proteins. You will get a different types. Suppose I am taking here many amino acids are there. Here I changed only one amino acid place. You can change the number of positions. That means you will get a different types of proteins. There are many types of proteins are there because of. So second one is the secondary structure. In the secondary structure, we will discuss about the what is the shape of the polypeptide chain. Already in primary structure, I explained about the it is explained about the sequence of the polypeptide chain. But in secondary structure, we will discuss about what is the shape in which the long polypeptide chain can exist. Suppose I will write one of the polypeptide chain A1, A2, A3 and so on AAN is there. It is one of the polypeptide chain. Okay, polypeptide chain. So already I said before that one. Here between the two amino acids, one bond is there. What is that bond name? Peptide bond. That bond is called as a peptide bond. Do you know what is the formula of the peptide bond? CONH. Do you remember this one? Already before that class I explained about the peptide bond. Okay, once you check that class, what is meant by peptide bond? In this peptide bond, CONH is there. CONH group. This is a amide group. This group is name is a amide group. That means between the two amino acids, what is the group is present? CONH group. That means here one CONH group, here also one CONH group, here also one CH. That means in this polypeptide chain, many CONH groups are there. Okay, that's what is called as a polypeptide. Only two CONH groups there means that is called as a, it can be tripeptide. Already said only one is there means there is a dipeptide because two amino acids are joined together. So here also many polypeptides are there. That means CONH bonds are there between the amino acids. So here one of the important point, the CONH. Here I already I have written one structure. This is a polypeptide chain. Here one CONH bond is there between the AA1 and AA2. Now AA2 and AA3, so you take another one CONH, another bond. Okay. Here also like that AA3 and AA4, one more CONH bond. These are called as peptide bonds. So the peptide bonds are there. Here one of the important properties there between this peptide bonds, again one hydrogen bond will form. How the hydrogen bond will form? Here nitrogen atom is there. Okay, nitrogen contains hydrogen atoms. Here carbon contains oxygen atoms. So because the oxygen and hydrogen form one bond, that bond is called as a hydrogen bonding. Okay, like that also. Okay, like that also many amino acids there. In this many amino acids, between the amino acids CONH bonds is there. So like that what happened, all CONH bonds form a hydrogen bond with next CONH bond. So because of what happened, actually we are thinking this is one of the longest straight chain, but it does not exist as a straight chain. It is converted into one spherical shape. Otherwise, it is converted into folding. It becomes folded structures you will get. Okay, there are mainly two types of folding you will get. One is the alpha helix folding, another one is a beta plated folding, that is called as a beta plated sheets. Because of this hydrogen bonding, these polypeptide chains can exist in two types of structures. What are the two types of structures are there? One is the alpha helix structure, alpha helix. Another one is a beta plated structure. Okay, do you understand why these structures are exist? Because of the hydrogen bonding. Hydrogen bonding stabilizes this alpha helix and beta plated structures. Okay, without there is no hydrogen bonding means we can consider it as one of the longest straight chain. So this is the alpha helix structure. See, in this structure, these are the foldings are there. Okay, these are the foldings. This is the alpha amino acids acts as a backbones. This is the one of the alpha amino acid CONH bond. Here also one CONH bond. Here also one CONH. Here also one CONH. Like this. If you take this fold now, in this one, to here CONH. Here also CONH is there. Here also in this folding also. This is the back side. In this back side also CONH. C1 is there. It is again come to the front side. It is also again CONH bond and CONH bond. It is again back side. It is a front side. Like this. The alpha helix is there. It is the alpha helix is a right-handed screw. It is a twisting to a right-handed screw. Okay, that means it is how this right-handed screw is there. Like that also this structure. That's what is called as a alpha helix structure. Comparing with right-handed screw. Okay, that I have to remember. So here what will happen? Hydrogen bonding will form now. Hydrogen bonding formed between the, here see this one, here also CONH is there, here also CO. In this back, this is back side, but back side CONH there means it does not form a hydrogen bond with in this one. Okay, this is the next turn we have to take. In this turn only again what will happen? It is come to the front side now. So both are front side is there. So because what will happen? In this NH bond, here CO bond form a hydrogen bonding. But here also CONH is there. It is opposite side. But here same side is there. So because of the same side, what happens? They form a hydrogen bonding. So again back side here is there now. One is the red color is the back side. It is also the back side. They will also form a hydrogen bonding. But here not given their bonds. Only front side is given. Here CONH. Here also CONH is there. Here also CONH. That means they form a hydrogen bondings. So because of the hydrogen bondings, it can exist as a alpha helix structure. Do you understand this point here? It is very easiest structure. You can easily understand. If there is no hydrogen bond means what will happen? It can exist as a one straight chain. But they will form a hydrogen bonding between NH and CO bonds. It is the one of the structure that is a alpha helix structure. One more structure is there. So this one is a beta plated seed structure. In beta plated, what will happen? Here two or more polypeptide chains you have to take. Before, in alpha helix structure, we are taking only one polypeptide chain. But if you see in this beta plated seed structure is there, in this structure, this is the one of the polypeptide. This is the one of the polypeptide. This is the another polypeptide. This is the another polypeptide. That means here many polypeptide chains are there. See this one, here given only 3 polypeptide chains. Here is a one amino acid is joined with another amino acid, another amino acid. This is one of the polypeptide chain. It is also another polypeptide chain. It is also another polypeptide chains. As the polypeptide chains are stretched out nearly maximum extension. They are very close to each other. This is the one polypeptide chain. The another one is there now. They are very closed. They are stretched very close to each other. Why they are stretching each other? Because there is a hydrogen bonding between this first polypeptide chain and second polypeptide chain. Suppose you are taking your second polypeptide chain, again form a hydrogen bond with the third polypeptide chain. Between this what will happen? Hydrogen bonding is there. That is called as a intermolecular hydrogen bonding. Everyone should remember in beta plated seed structure is there. No? In this structure, intermolecular hydrogen bonding is there. But before I said no, there is only one polypeptide chain. In this only intramolecular hydrogen bonding is there. But here we are taking the two or more polypeptide chains. They are stretched to nearly maximum extension. That's why. So we'll get a one beta plated sheet structure. Okay, why it is called beta plated structure? Because it is a folding, or these folds are similar to drapery. That's what it is called as a beta plated sheets. See, in our windows some curtains are there. The curtains also some folding is there. So same like folding also in this structure we will get. So because of that this structure is called as a beta plated sheet structure. It is also very easy. You can understand the structures. It is also hydrogen bonding, but what type of hydrogen bonding? Intermolecular hydrogen bonding. So this is about the alpha helix and beta plated sheets. Okay, now we will discuss about the tertiary structure. But what is the tertiary structure? Explain. Actually, it represents the further folding of the secondary structure. Further folding of secondary structure. Already we know in secondary structure two types of structures are there. One is the alpha helix, another one is a beta plated structure. Okay, alpha helix and beta plated structures are there. Again, this alpha helix undergo the further folding, means you will get a tertiary structure. Here also beta platelets are there. Beta plate also undergo the further folding, will get a tertiary structure. Already everyone know what is the structure of the alpha helix. So we are taking the alpha helix like this. This is one of the alpha helix. Okay. Suppose it undergo the further folding like this. Means this is called as a second tertiary structure. Like that also beta platelet structures also. In beta platelet structure also they undergo the further folding, means that is called as a tertiary structure. Here also again on further folding. So what will get? Two types of structures will get. Already we studied what is the structure of the proteins. Two types of proteins are there. One is the fibrous proteins, another one is a globular proteins. Do you understand this point here? In tertiary structure they undergo the further folding now. Because of the further folding, what will happen? You will get either globular proteins, otherwise you will get a one fibrous proteins. Everyone should remember there are two types of proteins you will get. Okay, so but which bonds are stabilized this secondary and tertiary structures? Everyone should remember mainly some forces are there. That forces are stabilized the secondary and tertiary structures. That forces everyone should remember. What are the forces are stabilize the secondary and tertiary structures? Secondary and tertiary structures. Okay, stability of the stability of secondary and tertiary structure. Okay, actually which forces are stabilizes the secondary and tertiary structures? I will write here. One is the hydrogen bonding. Okay, because of the hydrogen bonding they are more stable. Here one more bond is there. That is a disulfide bond. What is that one? Disulfide bond. That means between the sulfur atoms bond is that is a disulfide bond. Third one is weak van der Waals force of attraction. Van der Waals forces of attraction. That means between the molecules always attraction force is there. That is called as a van der Waals attraction forces. Fourth one is electrostatic force of attraction. Electrostatic force of attraction. That means there are many different types of attractions are there between this protein. That means between that alpha helix beta plated structures. So what are the main types of forces? Hydrogen bonding is there, disulfide bonding is there, van der Waals force is there, electrostatic force of attraction is there. Because of this all these attraction forces, so the secondary and tertiary structures are more stable. That means they can exist as either globular form, otherwise one sheet. That means fibrous proteins. Could you understand this point? Why That is, the secondary and tertiary structures are more stable. Means you have to write these points because of they form a hydrogen bonding. Okay, this is the one of the point. Second point is they also form a disulfide bonds. Third point, van der Waals. Do you know van der Waals force of attraction between the molecules? Some attraction force is there. So because of that, this also will get a more stable structure. What is meant by electrostatic force of attraction between the ions? COO minus ion, H plus ions is there. No? So some side chains also there. Because of that side chains also will get some electrostatic force of attractions. So these points you should remember in case of tertiary structure. Do you understand this tertiary structure? Next, we will study about the quaternary structure. Fourth one is quaternary structure. This quaternary structure explains about the what is the spatial arrangement of the polypeptide chains. What is the spatial arrangement? Everyone should remember. It explains about the spatial arrangement of polypeptide chains. Polypeptide chains. Because many proteins are made up of two or more polypeptide chains. Everyone should remember some proteins are there. No, proteins are made of two or more polypeptide chains. So that are called as a subunits. It contains two polypeptide chains. There is a two subunit. Three polypeptide chain means there is a three subunits. Suppose I will take one of the protein. This is a suppose I will take the secondary structure alpha helix. This is one of the I will take alpha helix like this. I will take I will take one alpha helix is there. This is the alpha helix. This is a further folding means what happen? You will get a secondary structure. This is the one of the protein. But some proteins contains two or more polypeptide chains. This is one of the polypeptide chain. Suppose I will take another polypeptide. Suppose I will take here like this another polypeptide chain. This is called as a second polypeptide chain. So these are called as a subunits. They are called as a subunits. This is one of the subunit. It is another means two subunits are there. These subunits spatial arrangement is explained by quaternary structure. The spatial arrangement. Okay. Do you understand this point? The spatial arrangement of subunits. Subunits is nothing but that is the polypeptide chains. Yet two polypeptide chains are how they are spatially arranged. That is explained about the quaternary structure. It is very easy to remember all these four types of structures. Primary structure explained about the sequence of the amino acid. Secondary structure explained about the what is the shape of this polypeptide chain. There are two types of shapes are there. One is the alpha helix, another one is a beta plated. Tertiary structure explained about the further folding of the secondary structure. Okay, but quaternary structure explained about the what is spatial arrangement of the two or more polypeptide chains. You should remember here take the two or more polypeptides that are called as a subunits. So this is about the structure of the proteins. In our NCERT book, one diagram is given. We will see the diagram now. So this is the NCERT textbook diagram. See, this is the primary structure. That means sequence of the amino acids. That colors indicate the what is the sequence. If you are changing the colors means you will get a different sequence. This is the easiest structure. Next, what will happen? Because of the hydrogen bonding, there is a folding will take place. So because of the folding we will get alpha helix structure. Here given alpha helix structure. Okay, either also will get some beta plated. This is called the secondary structure. This is indicated about the because of the hydrogen bonding some folding will take place. This is the secondary structure. Further folding of the secondary structure is called as a tertiary structure. It is the one of the secondary structure. Again it is the folding. Further folding means what happen? You will get a tertiary structure. So this is secondary structure now. Where continue that folding means you will get a tertiary structure. But in quaternary structure already said there you have to take the two subunits. Because some proteins are made of two or more polypeptide chains. It is one of the polypeptide chain. like that polypeptide chains you have to take two, I have to take, this is the one, I will take this one, this is the one of this one, another one is a this one, this is one, this is another one, that means two are there, that are the two subunits. It is a two polypeptide chains are there, that is called as a quaternary structure. It is very easy to remember the structures. Okay, primary, secondary, tertiary, quaternary structure. So this is about the protein structure of